Hello students, this is yesterday's lecture actually. Now we are moving on to see some development of systems in the last part of this course. So here we are going to look at the development of the cardiovascular system in two parts. In this first part, uh, we are looking at the development of the heart and how it gets partitioned, and in the second lecture we will deal with development of the great vessels, and the last lecture of this course will be development of the urinary system. So here we are looking at the beginning of heart development. Okay, so as early as the third week of intrauterine life, a cardiogenic area develops in the intraembryonic mesoderm. But originally, the cardiac progenitor cells arise in the epiblast, just like how the primitive streak had developed in the epiblast, and then they migrate through the primitive node into the intraembryonic mesoderm, right towards the cranial end, as shown here by that red area. This cardiogenic area lies even cranial to the buccopharyngeal membrane. That means it's right at the cranial end of the embryo and even cranial to the developing neural tube. So in this cardiogenic area, remember it's intraembryonic mesoderm. First they develop cell clusters, as you can see in the red stained area, and they are known as angiogenic cell clusters. These cells or clusters get together and acquire a lumen, and they form a horseshoe shaped plexus of small blood vessels initially. Okay, now this is a if in the figure, if this is the whole horseshoe shaped plexus, look at the most cranial end of this plexus, and that is the future cardiogenic area where actually the heart will develop. Also in the figure B here, you can notice, remember the lateral plate mesoderm got separated by a cavity called the intraembryonic coelom. Now note the location of this cavity in relation to the angiogenic cell clusters. Now figure B is actually a transverse section of figure A through the plexus. That's why you see the plexus on both sides, because it's a horseshoe shaped plexus, and you will see the cavity also on both sides. Okay, so this part of intraembryonic coelom, which is dorsal to the angiogenic cell clusters, will form the future pericardial cavity. Okay, so note these clusters are where now located in the intraembryonic mesoderm, specifically in the splanchnic mesoderm, which is the part of lateral plate mesoderm. You remember lateral plate mesoderm again got split into splanchnic and somatic. Okay, now in this lowermost figure, if you look at a sagittal section of the embryo, you can see the relationship of the developing pericardial cavity to the angiogenic cell clusters or the cardiogenic area. It is dorsal. Let's see what happens to it next. So in this angiogenic area, while some of those angiogenic cell clusters are forming primitive blood vessels, at this most cranial end, the angiogenic cell clusters are getting together and fuse to form first two endocardial heart tubes or two endothelial heart tubes. Soon, as you can see, these two tubes will start fusing from the center, and the fusion proceeds cranially and caudally until you get one single primitive heart tube or primitive endocardial tube. But the fusion, you can see, does not happen at the extreme ends of the two tubes. So we shall see how from a simple tube how the heart will develop into the adult heart. Okay, now folding of the embryo also has a role to play, because this embryo will fold craniocaudally and lateral folding, thus bringing all these two heart tubes together, and then the fusion happens. So sagittally, if you see the relation between the cardiogenic area, which is anterior to the oropharyngeal membrane, and the pericardial cavity initially is dorsal. But now when this embryo folds, as you can see in the third picture, because of the growth of the brain, the cardiogenic area, which was here, if in this picture you can correlate, most cranial, will come to lie down ventrally. And let's see what happens to the pericardial cavity also. So, as I said, what is the effect of craniocaudal folding of the embryo? You're seeing it in sagittal section. Now, here is the cardiogenic area. And remember, first the pericardial cavity was up and the cardiogenic area was down. Now, as a result of folding, there is a 180-degree reversal of relationship. That means the heart tube comes to lie ventral and the cardiogenic the pericardial cavity comes to lie below. Let us see in the next figure. So here, this is originally what is the situation before folding of the embryo occurs. So as I said again, heart tube, and above that or dorsal is the pericardial cavity derived from the original intraembryonic coelom. Now also note here this mesoderm. Remember, it's all splanchnic mesoderm around the developing heart. Now it is labeled as septum transversum. Now, as a result of folding, can you see the 180-degree reversal of relationship? That means the heart tube has come to lie dorsal now, and the pericardial cavity has come to lie below or ventral, and that is the septum transversum. Now, note in the third figure, again the complete reversal, and the heart tube in fact starts now invaginating or pushing into the pericardial cavity, just like how the lungs do into the pleura or any organ into the peritoneum. Is that okay? So note this relationship of each other. Now in a whole embryo, in relation to the whole embryo, here's where we are. So the heart, remember, was here, and now because of the folding it has come ventrally and it will now push into the pericardial cavity. Now the brown tissue here you can see is the septum transversum, which gives rise to many things, but one of the things that it will form from below, you can understand, is that the fibrous pericardium starts growing like that from below around the heart. Okay, and this septum transversum also gives, rise to part of the diaphragm. So in the adult, you will find, or you must have already seen during your gross anatomy practicals, that the lower portion of fibrous pericardium is fused below with the central tenon of the diaphragm. Why? Because they have both developed from septum transversum. Fibrous pericardium also forms from other sources, but we are not worried about that now. But from below, it is forming from septum transversum. That's why you see even the nerve supply is shared, right? The phrenic nerve supplies fibrous pericardium, also supplies the part of the diaphragm, etc. Now let's look at formation of the proper pericardial cavity and what is the transverse sinus and how it develops. Now this heart tube, see, after the folding the relationship has reversed. The pericardial cavity is down here, heart tube is above. Now this heart tube starts invaginating or pushing into the pericardial cavity as shown in section here. So temporarily this heart tube as if is suspended by a fold known as the dorsal mesocardium, just like a mesentery of the heart. Okay, so that will be here, or imagine a fold suspending it. But now this heart tube is growing very rapidly as it is invaginating the pericardial cavity. So it starts folding because the chambers are growing rapidly and it starts folding. You can see the arterial and venous ends trying to come closer to each other like a U-shaped loop. At the same time, it's pushing into the pericardial cavity. So this dorsal mesentery or dorsal mesocardium, which was here, starts degenerating, and that space left behind between the great arteries in front and the veins behind is known as the transverse sinus of pericardium. And look at the oblique sinus of pericardium will develop where the visceral pericardium becomes continuous with the serous layer of the visceral becomes continuous with the parietal layer of serous pericardium, which in the adult heart lies behind the left atrium between the four pulmonary veins. Okay, so this is the place of the oblique sinus, and transverse sinus will be here between the great vessels. Okay, so in short, how does the transverse sinus develop? It develops by the degeneration of the dorsal mesocardium, and then you have as if the heart is suspended like this between the various layers of pericardium. So this is pericardial cavity, serous layer with the parietal outside, the visceral layer, and that is the fibrous pericardium which has developed around it separately. So this is where we are. Here is another figure showing you the heart tube bulging into the pericardial cavity, folding, and hence the dorsal mesocardium degenerating, the transverse sinus developing. Also by now, this simple endothelial heart tube now gets surrounded by a thick layer of mesenchyme, which is splanchnic mesoderm itself, and this is known as the myoepicardial mantle. This will soon differentiate into the myocardium and the epicardium, or the visceral layer of serous pericardium. So in effect now the heart tube, even as it's developing, is made up of three layers: the endocardium, that is endothelium, then myocardium, and the epicardium, which is visceral layer of serous pericardium, and then the pericardial cavity, rest of the pericardium is surrounding it. Okay, so isn't it just like a blood vessel made up of three layers in the wall? Now this primitive heart tube here now is surrounded by the pericardium. As you can see, has a cephalic or an arterial end shown in red and a caudal or a venous end shown in blue, where the two ends have remained separate. Now the arterial end is continuous beyond the pericardium cavity with a little sac known as the aortic sac. We shall see about this in the next lecture. So let's see what happens to this simple heart tube now. As you can see here, it is already showing bulging, like some chambers are expanding, so parts are narrow in between. And remember, the simple heart tube, even at the third week, beginning of third week in fact, starts beating. That means blood starts flowing through even as it is developing and it is getting partitioned. This is an important point to remember. So what are the chambers as it is forming? Now we can name these chambers of the developing heart tube from caudal to cranial. So from the venous end, if you start, the first chamber is known as sinus venosus, or a venous chamber, which has two horns, a right horn and a left horn of sinus venosus, which then is continuous or opens into a common atrium. This opens into a common ventricle, and then the next chamber is known as bulbus cordis. Cordis means heart, bulbus means again swollen, which then is continuous with truncus arteriosus, because we are coming towards the arterial end, and that is continuous with aortic sac. So these are the chambers from below upwards. Now note in this figure itself that each horn of sinus venosus receives three kinds of veins: the right and the left umbilical veins, the vitelline veins, and the common cardinal veins. This is, you can only briefly remember that among the two umbilical veins, the right umbilical vein degenerates very early, so it's only the left umbilical vein that persists, okay, during fetal circulation, and of course after birth, you know, that degenerates too and becomes the ligamentum teres of the liver. The vitelline veins are nothing but blood vessels of the yolk sac, the absorbed part of yolk sac. Again, their fate will be seen when you do alimentary system. Now common cardinal veins are veins that bring venous blood from the fetus itself towards the fetal heart. So don't bother about them, but these are the three veins opening into each sinus venosus. Okay, now let's see what happens further to this heart tube. So you can understand from these figures itself that this heart tube shows rapid growth, but some chambers grow more rapidly than the others, and it's usually the common ventricle and the bulbus cordis that show more rapid growth. Hence the cardiac tube starts folding, as I said, first into a U shape and then it will twist, so it becomes like an S shape. Now because of this rapid growth of the common ventricle and bulbous cordis, they come to lie at the bottom because they are the heaviest, and this is known as the bulboventricular loop. Okay. So let us see now the fate of all these chambers and how they get partitioned. Now we will look at partitioning of each chamber in sequence as we go along, but remember it's all happening simultaneously. So the principle of cardiac septation is, or partitioning a chamber, is that when you have one chamber, you can try to partition it in various ways as shown here. So first way we can partition, you can think, is two ridges can grow from opposite sides of the chamber and meet in the midline somewhere, and then partition the chamber into two, or the space into two, correct? Now these ridges can grow from side walls or from the roof or floor, whatever. The second way in which we can partition is that from one wall, it might be roof, floor, or any wall, a single partition tries to grow and reach the other side. Now the third thing that can happen is a partition starts growing, say from the roof as shown in D, E, F and all that, towards the other side, but this chamber itself that we are trying to partition is expanding continuously. So you see, will this partition ever reach the other side of the wall? No, because the chamber is growing bigger and bigger. It's like you're bringing some partition down from the roof, but that roof is growing higher and higher, so it won't reach the floor. So in this case, what happens is we need some contribution from somewhere else to complete the partition. That means some cells from below at the same time can grow upwards to this downcoming septum and then add to the partition, right? So we will see how different chambers of the heart get partitioned in different ways. Now, the first thing to happen, the first stage of cardiac septation is with the development of endocardial cushions. So where are we going to construct these endocardial cushions? Now look at the model of the heart below. Imagine this lower chamber. This is a coronal section, right? As if you're looking into the heart by cutting it into a coronal section. So you can see the lower chamber is the common ventricle. The upper one is the common atrium. So this connection between these two chambers will be known as the atrioventricular canal. Now the first thing to happen is this partitioning of the atrioventricular canal into a right and a left AV canal. How does that happen? By developing of two ridges. So here is an example where two ridges grow from opposite walls, and these are known as dorsal and ventral endocardial cushions. Okay, they grow towards each other, fuse in the midline, and hence that AV canal is divided into right and left, which you can see here, right and left passages, or here also it's seen clearly. And this is the fused endocardial cushions. Okay, imagine with the other part of the heart also complete, this would have been like a ridge, right, going from anterior to posterior. So this is the first thing that happens. Now let us look at how then the atria are partitioned. So how this division of the primitive atrium happens into right and left atria by the appearance of septum primum and septum secundum. Let's see in the subsequent lectures. Okay. Now incidentally, this dorsal and ventral fused endocardial cushions is also called septum intermedium because it's coming in between, develops in between the common ventricle and the common atrium. Okay. So let us see now partitioning of the common atria or development of the interatrial septum. Now follow the figures. The write-up is for you to read, but you can understand from the figure itself. Now from the roof, this is shown here in the schematic diagram. This is, imagine, one common chamber or the primitive atrial chamber as we saw in the heart tube. Now from the roof of this primitive atrial chamber, a septum starts growing downwards, trying to fuse with the endocardial cushions. Remember, the fused endocardial cushions are here at the region of the AV canal. So that's the right AV canal and left AV canal has already been partitioned, right? Now this septum primum tries to grow towards the endocardial cushion. So the gap between the septum and the endocardial cushion initially is known as foramen primum or ostium primum. Okay. Now as the septum primum grows downwards towards the endocardial cushion, obviously this foramen primum will close. But remember, it's a beating heart and blood is already flowing in this chamber. So blood starts flowing from right to left side as this partition is growing, and it must continue to flow. So what happens? Now when this septum primum trying to reach the fused endocardial cushions, before it can fuse, a second opening develops in the septum primum itself, and that is known as ostium secundum or foramen secundum. Why must it open? Because blood must not stop flowing. So just before this fuses, this opens in the septum primum itself, and then the blood flows from right to left side. Now this foramen which opens in the septum primum up here is known as ostium secundum or foramen secundum. So where does it develop, or where does it open? It develops by breaking down of the septum primum in a superior area. Okay. So the heart chambers remain continuous. Now let us look at how the septum secundum develops and what happens to it. So while this septum primum is growing, then before it closes, the foramen primum closes, a foramen secundum opens higher up in the septum primum itself to allow blood to flow. Now simultaneously a second septum, so it's called the septum secundum, starts growing from the roof of the primitive atrium, where towards the right of the original septum primum. That means here it will grow here, as you can see, this light pink area, right? Now it tries to grow down, but it will not reach the endocardial cushions. It remains like a flap valve. And now the opening between septum secundum and septum primum is known as foramen ovale. This is like a flap-like opening, okay? And blood continues to flow through this flap-like opening like this from right to left. Now this is the last figure is actually a defect, so we will deal with this atrial septal defect later. Okay, this is something abnormal here, so don't worry about that right now. Hope you're understanding this process. Now actually see these septa. This is like a three-dimensional view. This is the fused endocardial cushions. So the blue is the septum primum that tried to fuse, but before it fused, a second ostium secundum or foramen secundum opened in septum primum higher up to allow blood to flow, and then a second septum, that is the green septum secundum, grew on the right of the septum primum, but it creates a flap-like opening like that, and that is the foramen ovale. Okay, and this is the actual embryo showing the breaking down of the septum primum higher up to form the foramen secundum. Okay, so actually these septa are semi-lunar shaped. So now let us see how these atria have got partition and how does the final atria develop. So again, first in the picture, endocardial cushion, that is the septum primum, the septum secundum growing, the foramen ovale flap-like opening has formed. Now let us see what happens to these primitive atrial chamber. Now because of this development of septum secundum and primum, the atria have got partition into right and left side, but they are still very small, right? So they need to enlarge. So how does the right atrium then finally form? Now behind this, because this heart tube is folded, remember, behind this is the right horn. See, like this. This is sinus venosus, as we said. This is common atrium. Now remember, while the heart is getting partitioned, it is also folding like that, right? So that sinus venosus and this atrium also has come to lie up here. This has come to lie down and all that. So behind the partitioning atrium is the sinus venosus, or here, after the heart has folded and twisted, etc. So the right horn of sinus venosus, that means here this right horn, starts getting absorbed into the right half of the primitive atrial chamber. Okay, and that is the blue that is showing the absorbed part. So this absorbed part, which represents the right horn of sinus venosus, now will become the smooth part or smooth posterior wall of the right atrium. And this original right atrium, or the primitive atrium, will remain as the rough part of, or the rough anterior part of the right atrium, which is represented by the right auricle in the adult. And you remember, it has the pectinate muscles, right? Now where is the junction between the two parts then, you know, rough part and smooth part? So that is here, the right venous valve, or which persists as the crista terminalis. This is the remnant of the right sinoatrial valve. Why sinoatrial? Because imagine, between the sinus venosus and common atrium, there are valves, right and left valves. So if this right sinus venosus is getting absorbed into the right atrium, obviously that valve will be at the junction of the smooth posterior part and the rough anterior part, which now is represented by the crista terminalis. And the right venous valve also will be represented in the adults by the rudimentary valve of inferior vena cava and valve of coronary sinus, means which are non-functional valves. Okay, so we'll see more of this later. In other words, now how has the right atrium developed? So original primitive right atrium is the right auricle, rough anterior part, and whatever is absorbed from right horn of sinus venosus becomes the smooth posterior part of the right atrium in the adult. So while this is happening in the right atrium, you can see this is getting absorbed. What's happening on the left side? Now even the left atrium needs to enlarge. Now in the left atrium, you have a primitive pulmonary vein already opening. Now the branching of this pulmonary vein is such that first there's only one single vein which branches into right and left, and then each right and left pulmonary vein has again two branches, because they'll be going to the lungs, right? Or they'll be coming from the lungs. Now this right atrium also tries to enlarge by doing what? By absorbing these primitive pulmonary veins into it. So first it will absorb the main pulmonary vein, then the right and the left. So each right and left then, you know, divides again into two. That means what is the end result because of the absorption of the primitive pulmonary veins is that 4 pulmonary veins individually open up then, and the left atrium has enlarged. What about the primitive atrium? That will again persist as the left auricle, or the rough part of the left atrium, and the smooth part has been derived by absorption of the primitive pulmonary veins. So this is the development of the atria. As you can see, the interatrial septa are in place, and flap-like opening, that is the foramen ovale, is formed already. So here is the summary of atria development. You can again try and identify all these things now. Now in this model, this is showing you behind, it represents the sinus venosus, right horn, and these are the right and left venous valves, or right and left sinoatrial valves. Okay, and on the left side you can see the primitive pulmonary veins getting absorbed. So try and identify all this. Now remember, this foramen ovale, we said, has been formed to allow blood to flow from right to left side. When you do fetal circulation, you will understand why. So basically the foramen ovale allows oxygenated blood that has entered the right atrium from the inferior vena cava to pass into left side. Why? Because there is no lung function, no pulmonary circulation in the fetus, right? So it's the umbilical vein that is bringing oxygenated blood from the mother, from the placenta, pouring it into the inferior vena cava, which is then entering, the right atrium. So from right atrium to left, there is oxygenated blood passing. Now this is a flap-like opening. Now at birth, what happens? The pulmonary circulation suddenly starts, so the pressure in the left atrium increases, oxygenated blood starts coming from the pulmonary veins, placenta shuts down. So this septum primum then gets pressed against the septum secundum, and there is a physiological closure which happens immediately after birth. Now the actual fusion, so it's like functional fusion, right? They just appose together or stick together, but actual fusion is not there. So the anatomical fusion or closure actually takes place gradually up to 2 months after birth, and the foramen ovale is actually closed, right? Now if it doesn't close, what will happen? So in the adult heart, here's where we are. Now you can correlate all that you have seen in the gross anatomy practicals. So here is fossa ovalis in the right atrium. That represents the original septum primum. The annulus or limbus of fossa ovalis will represent the septum secundum. So the foramen ovale was lying here between these two, right? But it's normally fused. So if there is an atrial septal defect or a patent foramen ovale, obviously it will be here somewhere in the fossa ovalis or in the annulus, depending on which septum is defective, or between them. Okay. So here are the right and left auricular appendages. This is right atrium open, so this is right rough part, and the smooth posterior part, their derivatives, and that is the crista terminalis, you can see, and continues with the valve of inferior vena cava, valve of coronary sinus. So what does that represent? It represents the right sinus valve. The left sinus valve, it's coming up in the next slide, actually just becomes part of the interatrial septum. So here is, if there is a patent defect, patent foramen ovale. Small defects may go unnoticed, but big defects obviously are known as atrial septal defects, and what will be the problems that can happen? So there will be mixing of blood. Okay, so think of what can happen to a large atrial septal defect, and obviously the treatment is surgical. Now let's see the fate of sinus venosus. So far we have seen what happens to the right horn of sinus venosus. So in this picture, when the heart tube has folded, look at the sinus venosus has come to lie posterior to the developing atrium, right? So you can see the right horn and the left horn. Now right horn of sinus venosus, we've already seen, gets absorbed into right atrium to form the smooth part of right atrium, right? And the original primitive right atrium persists as the right auricle in adult and remains as the rough part. Okay, and the junction between the two is represented by the right sinoatrial valve, which then is represented by the crista terminalis, valve of inferior vena cava, and valve of coronary sinus. Okay, so look at the right and left sinoatrial valves first. So that's the right, or it's also called right venous valve, left venous valve. So if the right, because this right horn of sinus venosus is getting absorbed into the atrium, right atrium, so this valve becomes the crista terminalis, etc. Now what happens to the left venous valve? It is, as this right horn is absorbed, the left venous valve or the left sinoatrial valve just fuses with the rest of the interatrial septum, and it's also called septum spurium. Okay, so it's then, not a separate structure. Now note, after seeing what happens to the right horn, note the left horn of sinus venosus. It's in fact becoming reduced in size. It doesn't enlarge like the right horn, and finally it will just persist as what? So look at this, it persists as the coronary sinus, which will end up opening in the right atrium itself, because that it is a sinus venosus only has been absorbed to form the right atrium, right? So the left horn persists as coronary sinus and opens into right atrium. Okay. Now let's look at how the ventricles are going to get partitioned. So I hope again from this model all the structures in the upper common atrial chamber and developing atria now you can identify. Now again get our focus. So this is the fused endocardial cushion and right and left AV canals. Now we are going to partition this bottom chamber or the common ventricle. So what happens is a muscular ridge from this myocardium wall of the common ventricle, a muscular partition, thick partition you can see, starts growing upwards towards where, towards the, or from where does it start growing? First it starts growing from the floor. So for the atria, remember these septa started from the roof, but here it's from the floor. Grows towards where? Towards the endocardial cushions. Okay. And you can see here this gap that is formed temporarily between the partitioning ventricles. This is known as the interventricular foramen. So this is the future right ventricle and the future left ventricle, and this is the muscular part of the interventricular septum, a thick part which has grown from the floor. Now this gap, that is the interventricular foramen, needs to be filled, but it will be difficult to be filled. Why? Because this is an enlarging chamber. Remember, the ventricle, the bulboventricular loop grows very large, so this partition can never reach the top here, because as it's trying to grow taller, but the ventricle itself is going to go bigger. Hence we need cells from some other source, and this gap, the interventricular foramen, then will have to be filled in by cells coming from other sources, which will be some from the endocardial cushions and the right and left bulbar ridges. We'll see where they are in a minute. And this part, upper part of interventricular septum, then will remain thin, and this is known as the membranous part. It will grow downwards towards the muscular part here to complete the chamber. So let's see it in steps again. So this is another model of the heart, and this is as if the right ventricle is opened up. So imagine this was this is the common ventricle getting partition. This is the muscular part of the interventricular septum growing upwards from the floor of the common ventricle. See, the actual shape is again semilunar. Okay, now it's growing towards where? Towards the endocardial cushions, which are represented in yellow. And here is the interventricular foramen that is formed. Now this septum will never totally close this chamber because it's an enlarging chamber. So at the same time, you can see these red and blue colored areas. These are known as right and left bulbar ridges. They start to appear in the bulbus cordis area. Remember that simple heart tube? Bulbus cordis was above the common ventricle. Okay, so remember, it's all happening simultaneously. These right and left bulbar ridges are trying to partition this higher chamber, that is the bulbus cordis. But at the same time, what happens, because they are close by, they say, okay, this partition will never reach the top, so we can contribute to close this gap as well. That means endocardial cushions and the right and left bulbar ridges will send cells downwards to close this interventricular foramen, and hence by their fusion, so these three population of cells will fuse here, and their contribution will form the membranous upper part. Now this is a thinner part, and it will close the interventricular foramen. Okay, so then if there are ventricular septal defects, means if the ventricular septum doesn't develop normally, in which part do you think will it usually the defects be? Obviously in the thin membranous part, because there are many contributions from many sources of cells. Sometimes then too many cooks spoil the broth, right? So then the defect may be usually in the upper part, not in the lower part. Lower part also can occur, but it's rare. Okay, so finally the membranous part will fuse with the muscular part. So let's see how it looks like after fusion. So this shows you the different contributions from different sources for the interventricular septum. So here, this pink area, this is the muscular part, thick muscular part of the interventricular septum growing upwards from the floor, and the colored areas, the yellow represents the endocardial cushions, and the right and the left bulbar ridges. So here is the interventricular foramen that was open, and you can see now in the second picture how these cells have contributed towards closure of the interventricular foramen to form the membranous part of the interventricular septum, which fuses with the muscular part. And what are the sources? So here are the listed sources, as we said. Now after this interventricular septum develops completely, what is the end result? The interventricular foramen is closed down, and it will shut off communication between the right and left side of the ventricle. Now the right ventricle will communicate only from the right, that is the pulmonary trunk. Left ventricle connects with the developing aorta, and the right AV opening now connects only with right ventricle, left AV opening with left ventricle. So this partitioning is complete. Now we will see later what are these bulbar ridges and what do they do to the bulbous cordis in a minute. First, let us see how it looks in the adult heart finally. So you have seen this specimens of the heart. Now you can appreciate that this is the thick interventricular septum. So this develops from the muscular part by myocardium proliferation from the floor. And can you see the thin membranous part? So now you know which cells have contributed to that. And where will there be an interventricular septal defect in case these two parts don't develop normally? So usually the defect is here in the membranous part. Is that fine? Now let's see. Remember, we spoke of the bulbous cordis. So we have partitioned the common ventricle. Now the chamber above that, if you recall, the next chamber in the heart tube was the bulbous cordis. Now the right and the left bulbar ridges, or they also called conus ridges, grow towards each other and fuse. So that common bulbous cordis is partitioned also into two, and what will it give rise to? It ultimately persists as the outflow tracts of the two ventricles. So on the right ventricle side, what is that outflow tract area known as? It is known as the infundibulum of the right ventricle, correct? And on the left side, when you leave the left ventricle up, what is that area called? That is outflow tract is called the aortic vestibule. Okay. So now you can see what is the fate of the bulbous cordis, which will form the outflow tracts. How? By being partitioned by the right and left bulbar ridges, forming what? Infundibulum of right ventricle and aortic vestibule of left ventricle. Okay. Now we have moved up. So we have partitioned the heart in the sense we partitioned the atria, we partitioned the ventricles, and we partitioned the outflow tracts. What remains next to be partitioned are the two big vessels. So what was that next chamber above the bulbous cordis? Remember the heart tube like this. So the higher chamber was known as truncus arteriosus. So as the right and left bulbar ridges grow, they also become continuous with ridges up here, and they are known as right and left conotruncal ridges. They are continuous below with the bulbar ridges. Okay. Now, but specifically in the region of the truncus arteriosus, these right and left conotruncal ridges which develop in the region of the truncus arteriosus, they will fuse with each other, but they not only fuse, but they twist around each other. See, can you see the twisting here? So first they start growing towards each other, but they form a twisting spiral septum. Hence, the truncus arteriosus then is divided into the ascending aorta and the pulmonary trunk, the immediate vessels that will receive blood from left and right ventricles specifically. Okay. So how do they get partitioned? By the spiral aortopulmonary septum. It's also called the spiral septum. Okay. And remember the bulbar ridges which gave rise, these are the right and left bulbar ridges. Below they gave rise to, or they contributed to, membranous part. Above they fused with each other to form the out and then divided the outflow tracts of the ventricles. Now still above they will fuse and unite with the spiral conotruncal septum. Okay, so one becomes like one continuous partition, and then you get the two vessels being partitioned. So here you can see this is the common chamber, but the spiral septum developing, the right and left conotruncal ridges fusing to form the spiral aorticopulmonary septum, and finally see the adult relationship, that's why, of the pulmonary trunk and the ascending aorta. Aren't they wrapping around each other like that because of the spiral nature of this septum? Now, another important contribution to this spiral septum is also by migration of neural crest cells. So you can see from head end they are migrating and they end up even in the spiral septum. We don't know the reason why. Maybe things that need to be in place soon, have additional contributions from there. So that means the spiral septum, or it's also called the aorticopulmonary septum, divides the truncus arteriosus into ascending aorta and pulmonary trunk. So there we have partitioned the whole heart, including the great vessels. So at the end we can just see quickly how now inside these chambers, walls, the valves will develop, the cardiac valves. So you know that you have the semilunar valves of the aorta and pulmonary arteries, the aortic and the pulmonary valves, and you also call the AV valves, that means the tricuspid and bicuspid valves. They develop in a similar manner. So remember, inside is all endothelium, right, or endocardium. So inside on the endocardium or in endothelial wall of the heart tube or each chamber, swellings develop like that, like in figure A. What does it consist of? A covering of endothelium with underlying loose connective tissue. Now gradually see they are becoming thinner like that as there is some programmed cell death, and they become leaf-like as in C there. Okay, and they will persist as semilunar valves. Now for the atrioventricular valves, the mechanism is similar, but then as they get hollowed out like this, they remain tags are with the chordae tendineae, and they get attached to the ventricular wall or papillary muscles by the chordae tendineae. So they are basically being hollowed out from endothelium. Okay, so this is the summary of cardiac development. Now I hope you can describe it in your own words. Don't try to memorize all that is written on the slide. Try to understand with the figures. Okay, and this is the steps that we have outlined. Okay, and lastly, from knowing how the heart develops, folds from a single tube, gets partitioned, each chamber getting partitioned. Now you can understand if something goes wrong, what all possible anomalies can occur. So this is just a general classification of cardiac anomalies. Either it can be anomalies of cardiac looping, means it doesn't fold in a precise manner. Then you might get like a situs inversus in inverted kind of positions, mirror image, where right ventricle may be on the left side, left or maybe on the right, or you might get the whole heart rotated to the right. That will be called dextrocardia. So anomalies detail you will learn next time, but we can just say anomalies of cardiac looping and position. Then you see how septa have developed. So anomalies of cardiac septation, atrial septal defect, ventricular septal defect, or spiral septal defects, etc. Anomalies of valves, so valve atresia, stenosis, or vascular anomalies, means anomalies of the big vessels, which we shall see some next time. Okay, so thank you for now. Hope this is clear. If not, you can post your queries. Okay.